Today, we're digging into some really incredible research that tackles one of the biggest questions in the fight against cancer. What if you could actually stop a tumor in its tracks just by flipping a single genetic switch? We've all heard that when cancer spreads, when it metastasizes, that's when it becomes truly dangerous. It's that spread that makes it so difficult to treat. But the work we're looking at today explores a potential way to stop that entire process right where it starts.
All right, let's get into it. To really get why this is such a breakthrough, we first have to understand the massive problem scientists are facing. Let's talk about a cancer's deadly journey.
So the focus here is on colorectal cancer, which is unfortunately really common. But the real danger, the part that makes it so lethal, is when those cancer cells travel and set up new tumors in the liver. That process, metastasis, is the biggest challenge for both doctors and patients.
So, knowing what was at stake, researchers decided to go right to the scene of the crime, that exact spot where the cancer first attacks healthy tissue. They were hunting for a clue, a kind of molecular suspect that could tell them what was driving this invasion. In this image, well, it shows us that precise location. Using a mouse model, scientists were able to zoom right in on what they call the invasion front, which is labeled TI here. This is the microscopic battlefield where the tumor, T, is actively pushing its way into the healthy liver tissue, L. This is ground zero for their investigation. And boom, this is what they found. When they analyzed all the activity right there at that invasion front, one particular protein just lit up. You can see it right here. The levels of a protein called CXCL8 were off the charts, way higher than anywhere else in the tumor or in the healthy tissue.
So this discovery led them to a really powerful idea. What if CXCL8 is acting like a molecular flare? A kind of signal that's just screaming, go, and fueling the whole invasion. And that, of course, led to the single most important question of this entire study. What happens if you can just turn it off?
Okay, so they have their prime suspect. The next step was obvious. This is where we get to the really cool part, the experiment itself, where scientists try to flip the off switch on that CXL8 gene. Now, to do this, they used a really clever tool called shRNA. The easiest way to think about it is like a genetic silencer. It's this tiny piece of RNA that scientists can design to hunt down one specific gene, in this case, the one for CXCL8, and just block it. It basically stops the cell from ever reading the instructions to make that protein. So, did it work? Oh yeah. This chart is basically a resounding yes. The bars show you how active the CXCL8 gene is. The control on the left is at a normal 100%. But on the right, after they used the shRNA tool, look at that. Gene activity just plummeted down to about 13%. The message was pretty much silenced. And the direct result of that is, if the genetic instructions are gone, the cell can't build the protein. And that's exactly what happened. The CXCL8 protein almost completely vanished from the cancer cells. The switch was officially flipped to off. Okay, so the gene is off, the protein is gone. Now for the moment of truth, right? Did silencing CXCL8 actually stop the cancer from being so aggressive? Let me tell you, the results were absolutely staggering. First up, they tested the cells in a lab dish. This chart is showing how many cells could invade through a barrier. And you can see the normal cancer cells on the left are super invasive, but the cells where they silenced CXCL8, they barely moved at all. It's a total night and day difference. Just to put a number on that, it was a 14-fold decrease in their ability to invade. Think about that. Without that one CXCL8 signal, the cancer cells were basically paralyzed. They lost their was dangerous superpower. But the real test is always in a living system. And when they put these cells into mice, the results were even more unbelievable. The regular cancer cells formed huge tumors, but the ones with CXCL8 silenced, well, you can see it on the chart. There was pretty much zero tumor growth. They stopped the cancer cold. The researchers themselves said it perfectly in their paper. They wrote that these modified cells have lost this tumorigenic potential. I mean, they didn't just slow the cancer down. They basically took away its ability to form a tumor in the first place. It's just, wow. So this is obviously a massive finding. But it brings up an even bigger question. Why did silencing this one gene work so well? And this is where we get into the really big picture and why this matters so much for the future of cancer treatment.
Well, it turns out that CXCL8 isn't just a simple invade signal. It's actually more like a master controller, a key that unlocks an entire network of support systems that the cancer cell needs to survive and grow. And this chart here shows us exactly how. By getting rid of CXL8, they accidentally shut down two other vital signals: VEGFA, which helps tumors build their own blood supply, and BCL-2, which is like an anti-death signal that helps cancer cells cheat death. Both of those lifelines got cut dramatically.
Now, I know this diagram looks really complex, but the main idea is actually pretty simple. See CXL8 right there at the top? Think of it like the general of an army. By taking out that one single piece, the researchers caused this cascading failure all the way down the chain of command. The signals for invading, for surviving, for building blood vessels, they all just collapsed. It was a total system failure for the cancer cell. Which leaves us with this final, really powerful question. This research has found a potential Achilles heel for one of the most dangerous forms of cancer. It suggests that a therapy designed to do just one thing, to block CXCL8, could completely dismantle a tumor's ability to grow, survive, and spread. And that is a truly hopeful idea in the fight against metastatic cancer.